Okay, so this is part of bicycle. You know, we try to put it together, right? That's what it is. Except it's harder than put together a bicycle because each part here, bicycle parts are basically pretty rigid, and proteins move. Proteins are like a jelly. They have dynamics and they have different shapes. And not random shapes, but they have different shapes. And how do you put it together? And that's a challenge. That is a challenge. And you know, if you put together, you try to understand. When you get the structure, you try to understand. Well, you know, what's the tricks in this structure, right? How does it function, right? I will say that MD simulations in the past decade or more, has been mostly focused on, in my opinion, interpreting experimental data from structural biology, meaning X-ray crystallographers solve the structure and we simulate it and we see how it might morph in one way or the other. So it's like if the crystallographer solves this bicycle, we will simulate it and we will find out that, oh, this pedal moves in a circular way and this wheel moves in a circular way and this thing can rotate and so on and so forth. And then we make a story. That's the best scenario. Like they solve the structure and we make some interpretation. Many of them don't particularly respect us because they think our work is a little bit derivative. Because when they solve this structure and we make some interpretation, which is useful, but they feel like we're dispensable, basically. So can we do better? Can we do better? Do we need to do better? I think we do, because many structures are very hard to resolve experimentally. The large biomolecular assemblies are particularly challenging, and in cells you have all kinds of environmental elements you cannot capture in your test tube, in your crystallization. For example, you don't have membrane. Okay, you can have something to mimic the membrane, but sometimes it's not good enough. And in cell, you know, maybe certain signaling assemblies are very sensitive to local environment. You have to have the exactly right concentration of everything, exactly right pH, exactly right electrostatic environment. How do you capture that in experiments? Sometimes, you know, often it's challenging. And in simulation, no problem. We can do it. We can do it. We can control these things. And can we do something useful to go beyond just be a supplementary tool? And that's sort of the direction of the effort, I think. Now, let's say protein folding. I talk about protein folding, right? The polymer chain folding into a well-defined shape. This is my colleague's work on villain headpiece. It will settle down in, I hope, in a well-defined shape. I think this movie is too long, but bear with me. It settles down and it's stable. It's like one tiny piece, one tiny part of the bicycle, right? This is just one tiny part of that bicycle. And ideally, we want to do it for every part of the bicycle, right? Fold everything into shape, then put it together. But we cannot do that yet, and this may not be the best way to go forward, because that's, first of all, we cannot do it. So Secondly, even if we can, whether that's the most efficient way to do it is questionable. But anyway, so for some proteins, for many proteins, we can fold it. This is a science paper by my colleagues in the short research. They folded 12 proteins in that one paper, and that was a great deal. But we don't have to fold every protein. We can just take experimentally resolved structure of these proteins and try to put it together. Because getting the shape of individual parts is not so hard. Putting it together, get to the big assembly structure is hard. Okay, so now I will, how much time do I have? I have only 15 minutes? I haven't even started. Yes. Okay, so, but I think okay, I will speed up. So this is the earlier work for like, it's sort of like the bicycle turn, right? Or the rotating of the bicycle. It's just like protein kinase got turned off. All right. We simulate that. We understand how this switch is controlled, this rotating, how it's controlled. This is what I call interpretive work, because we started from well-resolved X-ray structure, and we make it move, and we try to understand, what controls the motion, and what's the mechanism, and so on and so forth. This is my first work in this area. 3 residues, 2 years work, so that's, I have not been very efficient. This is 2009, published 2009, so I started working in 2007. I will skip up details. I just want to say that whatever results we had about that DFG flip, that switch off, we had a prediction that it can be controlled by pH. And we were lucky enough to convince Professor John Curran to test it in his lab. And very fortunate and very gratifying that we see a very nice pH curve from the measurement. And so this has an important point, is that simulations have to be validated by experiments. You have to be thinking about experiments all the time. At least I do. Okay, so this is a protein called EGFR. This is cell membrane. This is a cell membrane, and the EGFRs are these things, two-parted. One part outside of the cell, one part inside of the cell. It pick up message, chemical message from the environment of the cell, and translate in, and start this whole complicated wire, the circuit. So if this thing go bad, it's closely related to various cancers, and it's an important drug target in cancer therapy. And I try to understand why a single mutation, seemingly harmless, can be closely related to cancer. Here in this kinase, you have a single mutation, and it can, in the long run, cause cancer. And in our simulation, we show that this protein somehow is funny. It doesn't fold well. It doesn't have a very nice shape. And in crystal structure, it has a very nice shape, but in our simulation, it doesn't. And but if you introduce a cancer mutation, suddenly it becomes very nice. This is called stability chaos index. So wild type, chaotic but good. Cancer mutant, stable but bad. And then we in the context of its function, we realize that it's easier for the cancer mutants to form dimers and to have uncontrolled signaling. So it will made up it will make up signals. Even if it doesn't receive any external signal, it will pretend it receives a signal and keep going. So this is experimental validation again, and this is sort of, we're saying stability sometimes can be bad, and nature can be smart in putting out, use instability as a safety mechanism. Okay, then we talk about EGFR, not just this domain. We're talking about the whole thing. This is active state. This is inactive state controlled by this signal in terms of chemical entities. So it's like a puppet model, right? This controls these chains, these strains, and the strains controls the function of these kinases. So this was sort of we using, informed by simulation, we put it together. So now we are seeing chemical assemblies. That was a dimer. This is a tetramer, 4 of them, and this thing can keep going and have many members of them, forms a cluster. Now, I'm going to talk about something else. Again, the key word is biomolecular assembly. Here's how a drug gets assembled to its target or hit its target and neutralizes it. So I didn't know it's going to work, but when I had Anton to play with, I just decided to go for it and to see whether in simulations the drug molecule will really, do what it's supposed to do. And we leave everything free and just say, yeah, go for it. Let the physics guide you and see what happens. What happens is that after some trying, in simulation, you see it goes into the nice binding pocket. And this is, if you take some drugs, this is, generally speaking, what happens in our body. The small molecule hit its target and then neutralize this. Now this structure, if you compare with the X-ray structure, they when it's right, it's spot on. Sometimes it doesn't work, but when it's right, it's spot on. And we try to leverage this method in drug discovery, which I won't talk about. I think I'm gonna jump around, jump forward. Now that was a protein small molecule assembly. Now let's try protein assembly. It's harder, and this is modest. Only 2 of them. See whether they can go to the right shape. And just 2 small parts come together. Can we do that? In this case, we were successful. This is basically how it looks. Now, this structure, if you compare with, we did this blindly. We did not know the correct results. So it's just fortunate or unfortunate that the experimental paper came out at the same time, separately. And now we have a chance to compare it. So I would say we were pretty good in getting this one right? Assembly. So you can do small job. We put 2 together. Can we put more together? Now we are continuing this work, and now we put 4 domains together, 4 of them together. So these were the 2 I just talked about. Then now we have 4 of them together. And this is inactive, and now we have active model. But actually, we have doubts about this model. So this is part of the process. Sometimes you're just not right, and sometimes you have to go back and redo it, and sometimes you make mistakes. But if you're honest about it, it's still okay, right? So this is a model. May not be right, but that's what we are trying to do. These are unpublished work. The 2 proteins assembly were published work, and we know that's correct. And but we'll keep going. Right? This protein is called JAK2. It controls the production of red blood cells. And if it's overly active, and for some patients that's the case, then we will have too many red blood cells, and that will have pathological consequences. Okay, so now this thing is something even more early stage. This is the important protein called the RAS. It's one of the most important proteins related to cancer, and we built a model that to stack these. We think that when it's working, it stack up like a helix, like this. If you look from the top, it looks like a helix growing from the membrane. This is the base of the membrane. It keeps going up, up, up. Okay. Then it will recruit all the active proteins to it. It's like a signaling tower. It will recruit the other proteins to it and form nice structures, and they keep going. And that's why it can send out a very powerful and intense signal because there's a cooperativity. Now, if you look at our model from top, center is the core I was talking about, the helix, keep going this way, right? Horizontally going this way. And then it recruits all these proteins that's really doing the biochemical job. I really like this model. It's from our speculation, it's from a lot of simulation, because we speculate a model, then we simulate it. If it makes sense to us, we keep going, or then we go back. If not, we go back, and so on and so forth. So this is something we try to do. Is this correct? I hope so. I hope so. That would really make my day. But the important thing is that just like when in the double helix work, Watson and Crick put out this double helix model, and in the paper they wrote, I quote, "We propose a model," and we like to propose models, and enabled by Anton simulations and by other simulations, with Anton, without Anton, and we propose models, and models can be validated. And this is the end of my talk. And I was very fortunate to have my colleagues supporting me and educating me. And David Shaw made everything possible. And I will stop here.
Yibing.